Thank you, Marianne, for the introduction. Hello, everyone. My name is Darren Wan, and I'm a director from Production Support Department at GenScript. And I hope you enjoy the talks so far and enjoy a meal as well. And GenScript is a CRO company focused on life science. And today I'm going to talk about next generation bispecific antibody manufacturing, especially about how we achieve superior productivity and purity with our TurboCHO platform. Okay, so bispecific antibodies, or BsAb for short, they represent a transformative wave of antibody therapeutics. So by design, they can recognize and engage two epitopes or antibody together simultaneously. And so it's opened up in novel mechanisms of actions for treating complex disease like cancers or autoimmune disorders. So currently, over 700 therapeutic pipelines are running, and 75% of these pipelines are still under R&D or preclinical level. Here are some FDA-approved bispecific antibody drugs. As you can see here in the list, some of them share the same formats, but some of them do not. These formats may include BiTE, IgG1-based, IgG4-based, and the clinical potential is clear, which is reflected by the growing number of the approved drugs. However, the complexity in function translates directly to complexity in production. Producing these sophisticated molecules efficiently remains a significant production holder for the whole industry. Okay, so what makes BsAb production so challenging? Well, I think several factors come to play. Firstly, expressing multiple polypeptides into the cells often leads to like secretion issues, and sometimes potentially end-terminal miscleavages. And secondly, achieving a balanced expression of different chains is often difficult. So we know that often one chain is over or underexpressed, impacting the formation of the desired BsAb product. And thirdly, the imbalanced expression combined with the complex assembly, will generate a diverse range of products or produce the process-related byproducts. And these products may include like nitrogen mixing and aggregates, homodimers, half antibodies, mischain mispairing, and other antibody fragments. And separating these closely related byproducts from the desired BsAb, or we can call it POI, protein of interest, is a significant purification challenges. It's a very big challenge. And fourthly, the complex structure of the desired BsAb product necessitates a sophisticated and comprehensive quality control system and characterization methods. So, the byproducts and the POI may have large molecular weight, and they have slight molecular weight differences. So that's why it's just challenging. So to address these multifaceted challenges, GenScript has developed an integrated strategy built around our TurboCHO platform. So this is not just about optimizing one step, but about optimizing and streamlining the entire production process. This includes first, molecular design. So apart from our patented codon optimization powered by deep learning algorithm, we're also employing some best practice in industry to engineer the BsAb format to favor the correct sub-assembly of the POI and minimize the impurity formation from the very beginning. Secondly, expression optimization. So here I will take our transient expression system as an example. This involves iterative platform optimization of the host cell line and the expression vectors, our cell culture media, and the corresponding cultivation process. It also includes some crucial chain ratio optimization for different BsAb according to their molecular characteristics. And thirdly, purification optimization. So by developing a systematic and data-driven roadmap for purification, we can select the optimal chromatography methods for different kind of BsAbs. And these methods are based on the differences between POI and major byproducts. We have also developed a digital purification methods recommendation system based on our accumulated experiences and expertise. And finally, robust analytics. Our default analytic items include SDS page, HPLC, endotoxin. We also use LC-mass for identification of target products and impurities. So by implementing analytics at each step throughout the production workflow, we can monitor the quality of production and help us to decide the process decisions. Okay, so let's move on and look at these strategies more closely. First, to reduce mispairing issues, we are employing some best practice industry, especially some engineering techniques. Some of the designs can significantly help us reduce heavy chain and light chain mispairing. For example, so we can introduce knob-into-hole electrostatic steering mutations to the constant region of the BsAb to direct the heterodimer assembly. And we can also use the cross-map design. And we can switch to symmetric IgG-like BsAb design, single-chain BsAb, common light chain design, or in vitro assembly strategies to help us to avoid the light chain mispairing from the very beginning. And then let's delve deeper into the optimization at the expression level. This is one of the cores of our TurboCHO platform. So basically, we are employing a continuous improvement cycle driven by design of experiments, or DOE for short. So first of all, we continuously upgrade the host cell line by gene editing, and followed by multiple rounds of screening and adaptation. And then we use iterative process, which allows us for systematically optimizing the interplay between several factors in developing our in-house cell culture, cell culture media, feed media, and also the corresponding cultivation process. So the model-guided design and analysis approach allows us for rapidly enhancing the productivity of the upgraded host cell line and the overall expression platform. Okay, as you can see here at the top of the slide, over successive iteration from TurboCHO 1.0 to TurboCHO upgraded 2.0, we have seen dramatic improvement for the titer of control map, which has increased from 1 gram per liter in 7 days to impressive 2.6 gram per liter in the same time frame. And currently, we are developing TurboCHO 3.0, for which we are expecting even higher titer. And you can see at the bottom of the slide, so actually we also reached gram level titer for some BsAbs and MAbs of different species, with the highest titer above 3.5 gram per liter, which is impressive for transient expression. So another key element in expression optimization is to balance the expression of different constituent chains. So, as we know that even with the optimized platform, the inherent property of different chains can lead to imbalanced expression. So by adjusting the copy ratios, sometimes we can improve the character assembly of the POI, and it will lead to higher yield and simplify downstream purification. So in this slide, we show the example of monofab FC, and it has a RF mutation on HC1. So here, I would like to clarify a little bit here. We know that monofab FC is not typical BsAb because it just has one V region, but as an expression and purification process really resemble BsAb. So that's why I put it here. Okay, so let's move on to see the tests and the data. So as we can see, different ratios were tested. And we can clearly see from the bar charts and the SDS PGL, we can see some changes in the relative amount and the purity of the POI versus some major impurities, including the HC2 dimer, and aggregates. So which allows us to select the optimal ratio. In this case, it was 1.5 versus 1 for achieving higher purity early in the production process. And in the following page, we have more cases which demonstrate that the fine-tuning expression ratios can help us improve the balance between yield and purity. So here we have a symmetric bispecific antibody here, which has Fab and SFV linking to the FC backbone respectively. And it has a RF mutation on HC2. So we can see that when expressed at a ratio of 1 versus 1, we can see from the gel that there are some HC1 related impurities, include the half antibody and the homodimers. So by overexpressing the HC2, we can say that we can get more POI with higher purity after first step purification. Also, for these two symmetric BsAb molecules, we found that by slightly increasing the night chain ratio, that we can get final products with better balance of yield and purity. Okay, then let's move on to purification, which is the other core of our TurboCHO platform. So we know that the so-called one-fits-all approach just doesn't work for the diverse world of BsAb. But we still managed to develop a systematic roadmap for purification. This roadmap depends on the BsAb formats, broadly classify them to fragment-based BsAb and IgG-like BsAb, including symmetric ones and asymmetric ones. So for some simple molecules like the symmetric IgG-like BsAb and the fragment-based BsAb, as a major impurity during production are some aggregates. So some standard purification like protein A purification for the first step capture, followed by polishing purification like SEC, ion exchange chromatography, will suffice to remove the aggregates. But for some complex asymmetric molecules, as they often have some, multiple chain mispairing issues, so a tailored purification method is essential. And such polishing chromatography methods are often based on the differences between POI and major byproducts. So our chromatography selection strategy leverage the purification recommendation system I previously mentioned. So this system uses information including the BsAb sequences. For example, the affinity binding to some specific resin like protein A and protein L, and BsAb's format information, and some essential physical-chemical property differences between POI and byproducts. And it can allow us to select the optimal methods and the sequence methods for polishing purification. And these chromatography methods typically include affinity chromatography, ion exchange chromatography, hydrophobic interactions, size exclusion, and mixed model. So by this recommendation system, we can standardize the decision-making consistency and also to make sure that operation is consistent, minimizing some, variabilities by human. Okay, with all this said, let's look at how it works in practice with a couple of case studies. So here we have a heterodimer IgG with two distinct nitrins. One is lambda and the other is kappa. So after the first step, protein A purification by map select sure LX with pH gradient, and we can see from the chromatogram, the SDS page gel, and also the LC mass that we have the major impurities are some nitrogen mispairings, which means that the heavy chain were correctly paired, but combined with the wrong nitrins. And our system analysis recognized that the kappa nitrin can bind specifically to the MAC-select VL resin, while the lambda nitrin would not. So it suggested that we can use MAP-select VL resin for nitrin mispairing removal. So we just loaded the sample to the MAP-select VL resin. So after implementing this purification, based on the chromatogram and the SDS page, we can see that this POI and the byproducts were separated in either the flow-through or different peaks. And we sent those samples for further analytics. And we find that the peak 1 contains a POI, which has both the lambda and kappa chain, while the byproducts with only the kappa or lambda chain were found in either flow-through or the peak 2. And also the final product of the pooled peak 1 showed great purity. The purity is 98% by sec-HPLC, and with purity 90% by non-reducing SDS page. And the intact mass data show that we have successfully removed the byproduct. Okay, then let's move on to case 2. So case 2 is also a heterodimer. It's a heterodimeric IgG, and we were leveraging the differences in binding strength in this case. So after the first protein A purification, we can see that from the SDSP gel that the purity is not ideal, with only 60% purity by sec-HPLC. And the major impurities were identified as aggregates, HC1, HC2, HC1, HC2, LC, which were some LC missing byproducts with incomplete night chains. So our system analysis indicated a differential affinity binding of these different species to the resin map select VL. So the correctly assembled POI with 2 night chains would bind to the resin more strongly than the 2 other byproducts, which is easy to understood. So we note the eluate from the first step purification to the map select VL chromatography. And we can see from the SDS page gel that the POI and aggregates were successfully captured and eluted, while the night chain missing byproducts were found in the flow-through. So this means that the following purification was quite straightforward. So we just have to remove the aggregates. So we conducted a punishing purification with SEC. And finally, the final product showed high purity with 95% purity by non-reducing SDS page and 94% purity by sec-HPLC. Okay, then let's move on to case 3. This case, here comes an asymmetric BsAb with FAP and SAV linking to the FC respectively. And this case, we show that we can use the difference in PI to separate the POI and the byproducts. So after the first step purification, that we can find the purity is only like 35% by SDS page and 49% by sec-HPLC. And the major impurities were identified as HC1, LC half antibody and HC1, LC homodimers. As shown in this table, we calculate the theoretical PI of the POI and the byproducts. The PI of the POI is 8.27, and notably, HC1LC gives a delta PI of 0.7, and HC1LC homodimer gives a delta PI of 0.6. So the significance in the PI suggested that we can use ion exchange chromatography as an effective separation method. So we know the sample to CEX with salt gradient, and from the SDS page of the sample after the CEX purification, we can see it was successfully separated the POI with these major impurities. And the final product, we put these elution fractions to further analytics, and the purity data show that we have successfully improved the purity of this asymmetric BsAb. Finally, we have case 4, and this is a format and a sequence derived purification for 2 cross-map molecules with CH1 and Cl swapping. And after the first step of protein purification, we can see a lot of byproducts on the gel for these 2 molecules. And this includes the aggregates and some free light chains and light chain dimers, and also some light chain missing fragments. And after the system analysis, we find these products, including the POI and the byproducts, they exhibit significant differences in surface hydrophobicity, which suggested that we can use the hydrophobic interactions to separate the products. So we know the samples to the Higg chromatography, and based on the SDS page, it shows that we successfully separate the POI with these various byproducts quite luckily. And also, we put these elution fractions to further analytics based on the final, the summary table that we can see the Higg chromatography has greatly increased the purity of these AC eluate samples. And both samples showed purity over 95% by SDS page and SAC HPLC. So both case 3 and case 4 demonstrate that the importance of exporting some essential physicochemical difference like PI and surface hydrophobicity for us to select the optimal purifications to separate BsAb POI and its byproducts if the molecular weight difference are not very significant. Okay, so in conclusion, that we know that BsAbs offers huge therapeutic promise, but their production is also very challenging from design, expression, purification, and analytics. So GenScript, especially our TurboCHO platform, has offered an comprehensive solution by integrating the molecular design considerations and high titer expression platform, sophisticated purification and analytical platform to provide and to ensure reliable bispecific antibody production platform. With over 10 years in the development and production of antibodies, we have handled over 200 different bispecific antibody formats and successfully delivered over 7,000 BsAb projects. And GenScript committed to innovation to fully support the next generation antibody therapeutics. Okay, so, and last, please feel free to visit our website at www.genscript.com. You can find more information about our service and some technical documents there. And also, if you have time, please feel free to drop by our booth at 404. Thank you guys for listening.
We have time for one quick question.
Hi, just 2 quick questions. Do you know, like, over the time, how does this, they doesn't form any aggregate, these proteins that you are producing?
So I mean, over the time.
Like, yeah, how stable these,
Yeah, I would say that we have tried 7-day expression, and this antibody seems quite stable. But I think it depends on the specific sequence of the molecules. We cannot say that for every molecules, the stability still remains the same after extended expression time. So, but according to our developmental data, that 7 days should be quite okay for most antibodies.
okay. And the next is like, with this like 2 rounds of purification, what is the eventual yield? Like, do you lose a lot, like what you produce and then what you get, like yield wise?
I would say that the recovery rate after under the protein loss after different step of purification, right? So actually, I would say it also depends. Especially sometimes the difference between the POI and the byproducts are so similar that even we handle this protein very carefully, that still the protein loss could be over 50%. But sometimes we can have some lucky results with protein loss less than like 20% for the polishing purification. But generally speaking, that I think the average protein loss after each polishing purification should be about 50%.
Okay, thank you.
Thank you.
Okay, sorry, we're over time. We're going to have to move on. So you can address those questions at the booth. Let's go ahead and give the speaker one more thank you.
Thank you. Thank you, guys.